Introducing new antimicrobials, a follow-up video on antimicrobial resistance. Hello everyone, I hope you enjoyed my last video on antimicrobial resistance. Today I'm excited to share an update on new antimicrobials. The World Health Organization has released a comprehensive list of priority pathogens that need further research and antimicrobial development. Let's explore this list in detail. Priority pathogens are classified into three categories: Priority 1, Priority 2, and Priority 3. Let's delve into the Priority 1 level, which is critical. Here are the bacteria that fall into this category: Acinetobacter baumannii, carbapenem resistant; Pseudomonas aeruginosa, carbapenem resistant; Enterobacteriaceae, carbapenem resistant, ESBL producing. Priority 2 consists of bacteria that have become resistant to antibiotics. These include Enterococcus faecium, vancomycin-resistant strains; Staphylococcus aureus, which has methicillin-resistant vancomycin intermediate and resistant strains; Helicobacter pylori, which has clarithromycin-resistant strains; Campylobacter species with fluoroquinolone-resistant strains; Salmonella, which has fluoroquinolone-resistant strains; Neisseria gonorrhoeae, cephalosporin, and fluoroquinolone resistance. Priority 3, medium. The following bacteria have a medium priority level: Streptococcus pneumoniae, which is penicillin non-susceptible; Haemophilus influenzae, which is ampicillin resistant; Shigella species, which is fluoroquinolone resistant. WHO's latest report on antibiotics, 12 approved since 2017. A recent World Health Organization (WHO) report revealed that 12 new antibiotics have been approved since 2017. Surprisingly, only 2 of the approved antibiotics, namely lefamulin and veberbactam, in combination with meropenem, belong to new chemical classes. The majority of the newly approved antibiotics are derivatives of existing classes, including cefiderocol, a cephalosporin, and fluoroquinolone derivatives like delafloxacin, lascofloxacin, and levonatifloxacin, which was developed as the prodrug levonatifloxacin, as well as the tetracycline derivatives eravacycline and omadacycline. Assessing agents against WHO priority pathogens list. In this evaluation, most of the agents examined were authorized to deal with common syndrome-based indications, such as treating complicated urinary tract infections, complicated intra-abdominal infections, community-acquired pneumonia, CAP, and acute bacterial skin and skin structure infections. Cefiderocol, the only effective treatment for critical carbapenem-resistant Pseudomonas aeruginosa CRPA, carbapenem-resistant Acinetobacter baumannii, and carbapenem-resistant Enterobacteriaceae CRE. When it comes to fighting critical infections like CRPA, CRAB, and E, only cefiderocol has proven effective. Out of the latest approved agents, 5 are designed to tackle CRE, while 7 aim to address other high and medium priority pathogens as classified by WHO. Veberbactam, a comprehensive product overview. Veberbactam is a unique beta-lactamase inhibitor that contains a cyclic boronate moiety. When combined with meropenem, it is highly effective against Klebsiella pneumoniae carbapenemase (KPC) producing CRE. Veberbactam is specifically designed to target serine beta-lactamases, particularly KPC. When it comes to treating certain infections, it's crucial to note that Veberbactam may not be effective against Class B MBLs. Additionally, it does not activity against Pseudomonas aeruginosa or Acinetobacter species as a result of efflux porin mutations. Lastly, it's worth mentioning that the combination of Veberbactam with meropenem has been approved for intravenous infusion. Lefamulin, the first pleuromutilin antibiotic for systemic use in humans. Lefamulin, an antibiotic belonging to the pleuromutilin class, has been approved by the US FDA for treating CAP caused by certain susceptible organisms, including Streptococcus pneumoniae, methicillin-susceptible Staphylococcus aureus, Haemophilus influenzae, Legionella pneumophila, Mycoplasma pneumoniae, and Chlamydophila pneumoniae. While lefamulin has received approval for certain uses, it has also shown effectiveness in vitro against various drug-resistant bacteria such as MRSA, VRE, multi-drug-resistant Neisseria gonorrhoeae, and Mycoplasma genitalium. However, it's important to note that the clinical efficacy of these in vitro activities is yet to be confirmed. Lefamulin can be administered either orally or as an IV injection. Pretomanid, the answer to MDR-TB and XDR-TB. For adult patients with MDR-TB, multi-drug-resistant TB, that is unresponsive to standard medication, as well as XDR-TB, extensively drug-resistant TB, there is now a highly effective treatment option available in the form of pretomanid. This nitroimidazoxazine is administered orally, and when used in combination with bedaquiline and linezolid, it forms the BPL regimen. Pretomanid, a drug that inhibits the synthesis of mycolic acid, leading to a weakened cell wall, is similar to isoniazid in this regard. It also targets non-replicating microorganisms through the release of nitric oxide, which induces respiratory poisoning. With its approval by the FDA in 2019 and conditional approval by India's National Tuberculosis Elimination Program in 2020, it has become an essential tool in combating tuberculosis. A summary of recently approved drugs, including cefiderocol, a cephalosporin with wide-ranging siderophore, has received fast-track approval from the U.S. FDA for treating adults with complicated urinary tract infections and hospital-acquired and ventilator-associated pneumonia, HAPE-BAP. The European Commission has also authorized the drug for treating aerobic gram-negative organisms in adults whose treatment options are limited. Cefiderocol has proven to be effective against different molecular types of carbamidase-producing pathogens by transporting across the outer membrane of gram-negative bacteria and collecting in the periplasmic space. It has a wider range of activity compared to recently approved BL-lactam-BL inhibitor combination drugs, which are only effective against certain molecular types, such as KPC and OXA48. However, it is not active against gram-positive organisms. Recent drug approvals for infections caused by gram-positive bacteria. Contezolid, an oxazolidinone developed by MyQRx, has been granted approval for the treatment of complicated skin and soft tissue infections from gram-positive bacteria, including MRSA and VRE infections. Lascofloxacin is a fluoroquinolone with activity against gram-positive pathogens and respiratory tract infections. Lascofloxacin was approved for CAP, otorhinolaryngological, and other respiratory tract infections. Levonatifloxacin, alevonatifloxacin, IV and oral prodrug, which has a similar spectrum to lascofloxacin, was approved for skin and soft tissue infections. Zoliflodacin, a potential treatment for multidrug-resistant N-gonorrhoeae. The urgent need for effective antibiotics that can combat high-priority pathogens, such as Neisseria gonorrhoeae, has never been more pressing. This bacterium has become more resistant to third-generation cephalosporins and fluoroquinolones. Zoliflodacin, the first antibiotic of the spiropyrimidinitriones class, has exhibited potential as a viable alternative treatment. Its unique mechanism involves inhibiting type 2 bacterial topoisomerase by binding to a distinctive site that differs from fluoroquinolones. It has a low resistance frequency and has proven effective in treating N-gonorrhea. It has also demonstrated efficacy against some gram-positive and gram-negative bacteria. N-gonorrhea mutants resistant to zoliflodacin have not been observed, even when bacteria are exposed to combinations between zoliflodacin and antibiotics already in use, such as ceftriaxone, doxycycline, and gentamicin. Zoliflodacin does not show the phenomenon of cross-resistance with fluoroquinolones currently on the market. Zoliflodacin has no genotoxicity or bone marrow toxicity, unlike some other fluoroquinolones. It is less likely to cause hematologic toxicity and is a highly selective bacterial topoisomerase inhibitor. Exploring the challenges of Clostridium difficile infection, CDI, and potential treatment obstacles presented by CDI, a common nosocomial infection that disproportionately affects the elderly. Even with the use of antibiotics, many people face recurrent CDI, leading to significant health issues. Fortunately, there are promising treatments like ridinilazole and beztuximab that can help prevent relapses and improve outcomes. Ridinilazole, a synthetic bis-benzimidazole antibiotic, is highly effective in treating C. difficile infections with a distinctive mode of action. It blocks the cell division of the bacterium by attaching to the minor groove of DNA, resulting in a better clinical response compared to vancomycin. Ridinilazole has limited systemic absorption and a reduced effect on gut microflora, making it an almost ideal option for treating CDI. Exploring the advantages of using ridinilazole for CDI treatment. Ridinilazole is uniquely designed to target toxins A and B produced by the bacterium, thereby reducing interleukin IL-8 levels. This functionality may prove beneficial in alleviating gut inflammation in individuals suffering from severe CDI. Moreover, compared to vancomycin and metronidazole, ridinilazole is less likely to cause side effects, particularly in the gastrointestinal tract. Monoclonal therapeutic treatment has been researched to combat recurrent CDI. Bezlotoxumab, a monoclonal antibody targeting C. difficile's virulence factor, has shown potential in preventing recurrent CDI when used in combination with antibiotics. It has been approved by the FDA and produced no harmful effects in phase 3 trials. Efflux pumps, membrane proteins, and RPP resistance are mechanisms that contribute to tetracycline resistance in bacteria. These mechanisms weaken the interaction between the antibiotic and the bacterial cell, inhibiting protein synthesis and reducing the effectiveness of first and second generation tetracyclines. Mutations in the 16S RNA subunit can also cause resistance, but are less common. Third generation glycylcyclines, such as tigecycline and eravacycline, have effectively overcome resistances associated with tetracyclines. They are distinct from previous generations of tetracyclines because they contain a substituent in position 9 of the tetracycle that cannot be recognized by efflux pumps. Eravacycline has a fluorine atom and a pyrrolidine acetamide group in ring D at positions C7 and C9 respectively, while tigecycline has a tertiary amino group in place of the fluorine atom. Eravacycline is an effective antibiotic for treating complicated intra-abdominal infections, with impressive in vitro results against a broad range of pathogens, including resistant strains. Its use has shown no cross-resistance with other antibiotics and has not been associated with severe liver dysfunction. Clinical trials have proven its efficacy. Dilafloxacin is a fourth-generation fluoroquinolone that is FDA approved for treating acute bacterial skin and skin structure infections, ABSSSI. It is effective against many gram-positive pathogens, including those resistant to quinolones. S. aureus is the most dangerous pathogen worldwide, and traditional antibiotic-resistant pathogens like MRSA increase mortality rates and hospital costs. New antibiotics such as tedizolid, dalbavancin, oritavancin, and dilafloxacin have been developed to combat resistant pathogens causing ABSSI. These antibiotics offer an alternative to traditional treatments, which have limitations such as high levels of resistance, hospital costs, potential toxicity, and the risk of developing C. difficile infections. Dilafloxacin has lower MIC values against a broad range of gram-positive pathogens compared to traditional fluoroquinolones. However, fluoroquinolones have a history of adverse events, and both the US and EMA have imposed restrictions and warnings regarding their use. Dilafloxacin, like other fluoroquinolones, carries a boxed warning about potential side effects. Dilafloxacin is a promising antibiotic with a 60% bioavailability rate after oral administration. It is used to treat skin infections and has potential for respiratory infections. Clinical development showed mild side effects, but studies showed no association with heart disorders or phototoxicity. Further large-scale use is needed to determine the safety profile. Carbapenem-resistant Enterobacteriaceae (CRE) infections pose a significant global health threat, with high mortality rates and limited treatment options. These bacteria inhabit animal intestines and cause gut and urinary infections. Classic beta-lactamase inhibitors are typically ineffective, and new drugs are needed to treat gram-negative bacterial infections, especially CRE. Beta-lactams are antibiotics that can be rendered ineffective by beta-lactamase-producing bacteria. Combining a beta-lactam with a beta-lactamase inhibitor, BLI, can overcome this. New combinations with cephalosporins and carbapenems provide broader coverage and have been approved for treating complicated urinary tract and intra-abdominal infections. An example is ceftazidime with avibactam. New active compounds are needed to combat CRE infections, along with the appropriate use of antibiotics. Ceftazidime, avibactam has been shown to decrease mortality rates from CRE infections, but resistance cases have already been reported. A powerful combination of the carbapenemic antibiotic meropenem with vaborbactam has shown in vitro activity against enterobacteria producing KPC. Vaborbactam is a new inhibitor of beta-lactamases based on the structure of boronic acid, which strengthens the activity of meropenem. It selectively inhibits beta-lactamases, mimicking the tetrahedral intermediate of hydrolytic enzymes in bel-lactam antibiotics, preventing the inactivation of the antibiotic. Vaborbactam is effective against many class A and C beta-lactamases and carbapenemases, making it a crucial tool for fighting KPC. Vaborbactam combined with meropenem enhances the antibiotic's effectiveness against carbapenemase-producing strains of Escherichia coli and carbapenem-resistant K. pneumonia. The combination is not effective against Acinetobacter spp or P. aeruginosa. Vabomere is FDA approved for complicated urinary tract infections. Ricarbrio, a combination of imipenem and relebactam, is a well-tolerated treatment for complicated infections such as urinary tract infections and intra-abdominal infections. It could also be a potential option for carbapenem-resistant Enterobacteriaceae infections, but further clinical monitoring is required due to limited extended clinical data. Plazomicin, a new aminoglycoside, has also demonstrated promising results in treating infections caused by multidrug-resistant Enterobacteriaceae. Plazomicin is a new aminoglycoside antibiotic designed to target Enterobacteria spp and block most of the aminoglycoside-modifying enzymes, which makes it effective against antibiotic-resistant bacteria. Unlike traditional aminoglycosides, plazomicin is not readily inactivated by enzymes, thus offering a promising approach to treating resistant bacterial infections. Cefiderocol is an effective cephalosporin antibiotic that combats community-acquired bacterial pneumonia, ventilator-associated bacterial pneumonia, and complicated urinary tract infections. It belongs to the fifth generation of cephalosporins, which are effective against MRSA and MDR bacterial strains. Ceftobiprole and ceftolozane are two other fifth-generation compounds used to treat community-acquired pneumonia and carbapenem-resistant Enterobacteriaceae and P. aeruginosa. Cefiderocol has a unique structure that provides stability against a wide range of serine and metallo-beta-lactamases, making it less likely to be affected by resistance development compared to other cephalosporins. It has a broad antibacterial spectrum, including against carbapenem-resistant strains, and shows potent activity against multidrug-resistant gram-negative strains. Multidrug-resistant tuberculosis MDR-TB has a high mortality rate and is challenging to treat, posing a significant risk to public health. Bedaquiline and delamanid are newly approved drugs for treating multidrug-resistant pulmonary tuberculosis MDR-TB. Bedaquiline, marketed as Certuro, introduces a new mechanism of action that blocks the ATPases proton pump that provides ATP to the mycobacteria. The role of delamanid and pretomanid in tuberculosis treatment. Delamanid and pretomanid share some of the same characteristics, including their lipophilic nature, which allows them to penetrate the mycobacterium wall. Pretomanid is currently available in tablet form and is effective in treating pulmonary MDR and XDR tuberculosis. However, it does not address extrapulmonary tuberculosis, a rare form representing only 5% of existing tuberculosis cases. Polymeric lipid nanoparticles are being studied to break down bacterial biofilms and combat antibiotic resistance. Rhamnolipids and polymer nanoparticles are being used to destroy the extracellular matrix in biofilms, making bacteria susceptible to antibiotics once again. The research aims to address the challenge of antibiotic resistance in Helicobacter pylori bacterial biofilm. A new antibacterial system uses a chitosan polymeric core encapsulating clarithromycin to fight bacteria, prevent biofilm formation, and bacterial adhesion. Silver and iron oxide nanoparticles coated in rhamnolipid also effectively eliminate biofilms of Staphylococcus aureus and Pseudomonas aeruginosa. Thank you for taking the time to listen to me. I hope you found the topic engaging and informative. Don't forget to like and subscribe for more great content.